So welcome everyone. So today we have the pleasure to have with us Professor Inés Armendáriz. So Inés is a professor in the Department of Mathematics at the University of Buenos Aires. And she is a member of the probability group in the same probability, in the same university, sorry. So in this way, her main research areas are related with probability and stochastic process. So today, Inés is going to talk about grouping testing with nested pools. So thank you again, Inés, for accepting our invitation. And it's up to you right now. Thank you.
Okay. Please, as I told Andresa, interrupt me at any point if you have any questions. And today I plan to talk about testing, group testing, and in particular some work we did together with my co-authors. Here we have Pablo Ferrari. Some of you may have met him. He had a very long career at USP in Sao Paulo. Then, and he's at the Universidad of Buenos Aires right now. Also Daniel Fraiman, who's at the Universidad San Andrés in Buenos Aires as well. Mario Martínez, who's at Unicampi. And finally, Silvina Ponce Dawson, who's in the physics department at the Universidad of Buenos Aires. And okay, let's start by one example, one of the first examples of group testing. This problem goes back to 1940s during the Second World War, when the United States were conscripting men for the US Army. And at that point there was syphilis, but it was not such a common disease. So, okay, they wanted to screen to see whether the men that were conscripting had the disease, but it was very expensive to test each of them individually. So Robert Dorfman was a statistician at that point, but then he would become an economist in Harvard. But at this point, he was working as a statistician, and he proposed a very simple method, which was the following. So instead of testing each potential soldier individually, he proposed to take blood samples from a group, which we will call a pool of soldiers, M soldiers. Then we will see what the value of M should be. So you take all the samples from this group of potential soldiers, you mix the samples, and you test them all simultaneously. You perform just one single test of all these combined blood samples. And then two things can happen. If the test comes negative, then all these soldiers have been cleared. You know that they're all healthy. And on the other hand, if the test comes positive, then on a second step, you test all of their samples, all of their blood samples individually to determine their status, whether they are infected or not with syphilis. And well, there are several assumptions here. First of all, we assume that P is known, otherwise one has to estimate it, where P is a probability that a single soldier is infected. And the second one, and this is important, we're assuming that this is noiseless in the sense that there are no factors such as dilution, for instance. One thing that would probably happen is that as you combine many samples, if there are many samples in the pool, then the dilution plays up, and then what happens is that you cannot detect the disease anymore. But we're assuming that this does not happen, that there's no limit to how many samples you can include in one pool and test it, and still the result would be accurate. It would be able to tell us whether one of them, at least one of them, is infected. And also we will assume throughout the talk that there are no false positives and false negatives. All these assumptions, actually, obviously one has to test them and see if they work. And in that case, one has to introduce some modifications to the models to allow for that. But we will assume here that none of them happen, so that there are no dilution effects, there are no false positives, and there are no false negatives. Tests are conclusive. Okay, so the goal here is to minimize the number of tests. And the way we're gonna check for this is by computing, so this is random, because we assume that each soldier can randomly be infected or not with probability P. And so the game is to reduce the number of tests, and we will measure this by computing the mean number of tests, and then divide by the size of the population. And since we are grouping them in groups of size M, so it's enough to say, okay, so we're studying them in groups of size M. So let's divide by M at the end to find out how costly this is in terms of each individual in the group. This, I hope this is clear. Otherwise, just okay, I'll write the formula now, so maybe it's clearer. So okay, let's compute this. So there are two stages. The first stage is when we perform just one test of everyone, these M individuals, and then we will move on to a second stage where we test each of the individuals separately, only if this first test comes up positive. So let's compute the probability that the test is positive. Well, the test will be positive, as assuming this noiseless assumption, if and only if there is at least one infected individual. So the probability that there is at least one infected individual is 1 minus the probability that they are all healthy, and the probability that they are all healthy is 1 minus p. P is the probability of the infection, raised, we're assuming they're independent, raised to the number of people in the blood samples in the pool. So we get this quantity here. Okay. Oops, sorry. And so this is the probability that the first test comes up positive, and then one needs to move on to the second stage. And in the second stage, we will simply just test everybody from the pool. So the mean number of tests in the second stage will be m, because we're performing m tests, times the probability that we do get to the second stage, and the second stage we get to it with probability, the probability we computed before, this one here, if we call q equal to 1 minus p, to make it shorter. Okay. So we get that the probability, the cost, I mean, here in the numerator, we have the average number of tests, which is 1, that's the first test, and then 1 minus q to the m is a probability that we move on to the second stage, and then we have to test m samples. So we get this numerator, and then we divide by the number of people that have been, whose status has been reviewed by this procedure, which is m. So we end up with this quantity here. So this is what Dorfman proposed back in 1943. And then one, let me just say, because this is the game, then one would try to optimize, I mean, P is data, this is something you know, and then what you try to do is to find the optimal M. And the way you think about it is that, well, the first intuition would be take M as large as possible, but then this is not informative, because if M is too large, then it's very likely that there will be one infected individual, and then you will end up testing everyone individually, and on top of that, you will have wasted your first test, because in the end you have to test everyone individually. So there's a compromise here. You want the first pool to be as large as possible, but not that large that almost surely you will get to a second stage. So you have to play with that. So there are many applications for this. You can think of many. The reason we started working on this was not our, none of us was really an expert on this, but the reason we came to this was because of COVID. And this was back in 2020, and a group of people, a group of biologists in the state of Santa Fe here in Argentina wanted to apply this. At that point, Argentina had very few tests that were not enough. It was not easy to get tested. And there was a problem that people in care homes and such were high risk. So there had to be some way of determining whether the people working there were infected or not. And so they proposed to use this kind of methods, and they contacted us so that we could help them with that. And that's how we got interested in this. So going on with the history of the problem, Sorel and Grohl, a little bit later in 1959, proposed to do the same idea. I mean, just work a little bit more on Dorfman's idea, and instead of applying it once, just try to do this recursively. So test the first pool. If it's positive, then how do you split it up? And what do you do to go on studying it, splitting it up, but without going to a second stage where you have to test everyone individually? And I will just discuss one of these methods, which is the binary splitting algorithm, which is very effective. It's kind of optimal in terms of cost. And the binary splitting algorithm is something very simple. I write the algorithm, but let me just tell you in words very briefly with this. You start with your pool. Suppose it's even. We will suppose that everything is even, so that we can split in half. If not, you have to introduce some modifications, but let's not worry about that. So you start with your pool, you test it. If it's negative, well, you know that every individual in the pool, every sample in the pool is not infected, so you take them aside. If it's positive, on the other hand, you split it in half, You take your first half and you test it. If it's negative, you know that all these individuals are healthy, you put them aside, but you also know that since there was some infected individual in the sample, then this individual must be on the other half, the one you have not yet tested. So you take the second half and you split it in two, and you keep repeating this. So let me just describe this here. One pool with n samples, you perform a single test on the pool samples. If it's negative, this is the case when everyone is cleared. Otherwise, if your pool consisted of just one person, then you know that this person is infected, stop. Otherwise, pick half the samples and to form a new pool and test it. If it's positive, then go back to the previous step and repeat. So take this half and split it in another and half itself and keep on going. If it's negative, then you know that this was all healthy, put them aside and start with the other half you have not yet studied. So just as we described, and here we have an example of what can happen. We start with, so here we have eight samples, right? So we have 8 samples in the pool, we test it, it comes up positive, then we split it, we take the first half, so we take this, we test it, it comes up positive again, fine. Then we take this first half, we test it, it comes up negative, so these 2 individuals are healthy, we put them aside, and we know that since we knew that someone was infected here, we know that there must be an infected sample in the pool, so we know that someone is infected here, so we don't need to test it, this information we already have it. So we take, say, the first sample, we test it, and we get a positive result. And then by doing this, we have information. Notice that when we finish this first leg of the procedure, what happens is that we have information, in this case, for this particular realization, we have managed to reveal that these 2 individuals are not infected, and this individual is infected. And then we still have 5 samples of which we know nothing. So there are 5 samples that yet need to be studied, so we have to start over now with an initial pool of size 5, and of course when we split it we'll have a pool of size 3 and one of size 2, but more or less the same idea. And so we need to start over with a new pool with everyone whose status has not been revealed yet. Okay, so this is the method. And one thing you can quickly, if you look at this, if you think about it, the fact that at each stage you're dividing by 2 means that it takes you order more or less, with some corrections depending on n really, but more or less the depth of this procedure is log base 2 of n, where n is the number of samples we had in the initial pool, because we're dividing by 2 at each stage. So this is how long it takes you to get to when you end with this procedure. Either you have cleared the whole pool, or you end up with one infected individual, such as this. By design, this is what happens. So you end up with one infected individual, then you start over. It will take you at least as long to get to another infected individual, and you keep going. So you get that, and each time you're going to be performing this at most this many log 2n tests. So by the end of the procedure, you conclude that in order to study, to find all k infected individuals, suppose that you have k infected individuals, in order to determine, to find out who they are, you need to apply this algorithm. I'm sorry, you need to apply this algorithm several times, but k times precisely. But the total number of tests you will need to perform is of this order, k log 2n plus some correction. Okay, and this is something easy to see. And now you can play around with the size of the initial pool. You need not test if you have a very large population, size n, and you've gotten all the samples. Say you're testing with the whole population in a hospital, everyone working in a hospital, and you get all these samples simultaneously. You need not start with an initial pool having size n, because as I said before, this might be useless, because you know that with probability almost 1, it will come up positive. So you wasted this test. So maybe it's best to split it into smaller pools and start with the smaller pools and do this all simultaneously in parallel. So the way to go actually is to start with initial pools of size 1 over p, where p we assume is known, is the probability of infection. And by doing this, if we call capital K now the number of defective items, which is random for us now, then applying this theorem, we find that we can we can reveal all the defective items in K, capital K, this is random, log 1/p. 1/p was the initial size of the pools, we split the initial population in, plus some correction. And now if we assume that we're in the situation where each individual is infected with probability P independently, we get that if we take expectations here, then the only, to first order, when we take the expectation of this term, that this is the only random variable, and the number of infected individuals should be, in mean, it's n times p, because they're independent and only random variables. So we get this, and then when we consider the cost, which is taking the mean number of tests and dividing by the size of the population, so we divide by n, we end up with this point. And now, this is the main message. This is the first order is P log 2 1/p, and this is the cost of this test. And in fact, it turns out to be excellent. I mean, this is almost as good as it gets. So in terms of cost, this algorithm is very good. And I included some references, and at the end I have a list of the complete references in case you're interested. So let me just try to convince you, or to convey the idea of why this P log 2 1/p is good. The first computation you can do to check this out, I mean, this is very simple, elementary computation, is in terms of combinatorial testing. So we have probabilistic testing and we have combinatorial testing. Probabilistic testing is what we've been talking about so far, which is when the number of defective items in the population is random and you don't know where they are, which ones are defective. And combinatorial testing, on the other hand, is when you have a population and you know how many defective items there are, and the problem is to determine which ones are defective. So in combinatorial testing, suppose we have n items, we have k defectives, k is fixed now, not random, and we're going to perform t tests. T is the number of tests. So yeah, and each of these tests will be, as in what we were considering right now, positive or negative. So this test, each test has a binary result. So the total combination of tests will be 2 to the t, right? Each of them can be positive or negative. On the other hand, the problem is to determine the defective set. The defective set, and there are the total number of n, the population, choose k possible defective sets. So assuming that you can choose any defective set equally, and each of the tests you're performing should be able to distinguish between these different choices of defective sets. So we should have that 2 to the t has to be greater than or equal the possible choices of defective sets. So we get this bound here. In order, if what the strategy we're designing, if we want the strategy we design to be good enough, we need to design the test so that they distinguish between the different possible choices of defective sets. So we need 2 to the t to be greater than or equal n choose k, and then using some elementary bounds for n choose k, we conclude that I'm taking logarithm and so on. That's why the log base 2 appears. We get that t should be greater than or equal k log 2 n divided by k plus some correction in k. All right. So we see that the number of tests in this combinatorial deterministic setup should be at least k log 2 n over k. And now if we think that k is p times n, more or less, that our choice of the number of defective items is p times n, and we divide by n, we bring it to the other side, we get back to this bound we had before, that the average number of tests per individual should be bounded below by p log 2 1 over p, which is precisely the bound we had for binary splitting. So this is very good. This is just for deterministic testing, but if we move on to the probabilistic testing, we get the same bound just using information theory, because the idea here, just in case maybe you're not familiar with information theory, but the idea is that it's more or less the same as before. The test strategy one is applying should be able to convey all the information of the infected variables. So if we think that the infected variables that we have this vector x, x1, xn, where each xi is Bernoulli p, indicating xi is just telling us the status of the ith individual. So xi is 1 with probability p if this individual is infected, 0 otherwise. And the test strategy should be able to tell us all the information we need about this vector x, because it should tell us which of these individuals are infected or are equal to 1 and which ones are not. So in that case, information theory tells us that the mean number of tests should be greater than or equal the entropy of the variable, of the vector x, and the entropy of the vector x is n times, since x1, xn are independent, is just n times the entropy of x1, and then one computes that, and we get this bound, that the expected number of tests should be greater than, this is the entropy of the vector x, and now you can check easily that, well, this is essentially the same thing as log 2 1 over p is more or less like p. So this second term is small relative to the first one. So this is the dominating term here. And once we divide by n, we get back to our order. So in any case, we see that p log 2 1 over p, it's an excellent bound. So binary splitting is very good in terms of cost. On the downside, I should say, so why not apply these? On the downside, one problem with binary splitting is that the number of stages is random, because you know that in log 2n, little n, you will find one infected individual, and then you will have also determined that a random number of individuals are healthy, and then you will have a pool of people you know nothing about, and you have to start over. So once you start out, and you cannot do this in parallel, so you have to finish with your first investigation to start with this, with what happens with this new pool you have formed. So the consequence of this is that the number of stages is random. And this, if you're running it and each test takes you some time, such as, I don't know, to do PCR, for instance, takes a little bit of time to process the information. So this could be lengthy. And that is maybe not that great. So we together with my co-authors here, we decided to try something else. And then, of course, we found out that what we were trying had already been discovered, maybe proposed many years ago by Johnson, Kotz, and Wu in 1991. But still, we could do some work because we could optimize. I mean, the game here is you propose the algorithm, and then you start optimizing in the parameters of the algorithm. So, okay, let's just go to the nested pooling scheme. The word nested here means that at each stage we will split. What we will do is that the pools at one stage will be sub-pools of the previous pools. So this is different from binary splitting, because when we finished with one investigation, when we were going through this branch and we finished, then we had one pool which was formed by leftovers, and we put everything together. So this was not a sub-pool of any of a pool at the previous stage. So what we propose here is nested in this sense, and the idea is absolutely simple. So we will do what Dorfman proposed to do just several times. We will do it several times. So we will have k plus 1 stages. We write it like k plus 1, meaning that the last stage, the plus 1 stage, means when we get to individual testing. This is the last stage where you test everyone you don't know anything about yet. So you determine the status of these individuals just by individual testing. And then we have so this is k plus 1 is equal to 1. And then we have m1, m2, mk. These are the sizes, the successive sizes of the pools. And we will assume that mj can be divided by mj plus 1. That mj is a multiple of mj plus 1, so that we can take the pool at the stage j, which has size mj, and then we can divide it into subpools of size mj plus 1, all equally sized. So the choice here is k and the pool sizes. This is a vector of pool sizes. And then we suppose we have arrived at the j stage, and we have one pool of size mj. We test it. If it's negative, all the individuals, this is just as usual, the individuals are cleared. If it becomes negative, and if the combined sample test is positive, we split it into mj over mj plus 1, pools of size mj plus 1. And then when we get to the end, everyone who has not yet been discovered is tested individually. So let's go to an example because it's much easier to see it in a picture. It's really simple. We start with a sample with a pool with n samples, where there is one infected sample, which is the one in red. So we test it. This is positive. We move on to the next stage, and we split it in 3 pools of size 3 each. We tested. This comes up negative, so we put them aside. The second one negative, we put them aside, and this one comes up positive. And then in the next stage, we just test everyone individually and we finish. By doing this, at the end, we know that we have determined the status of everyone in the initial group. So, okay, what's the cost of this? The argument is completely similar to what we did for Dorfman. Just repeated use of the same idea. So we assume we would call T of X, as before, the number of tests required to reveal the status of all items. X here is a vector, it's x1, xm1, so it's just a string of zeros and ones telling us the status of each individual. And each of them are independent Bernoullis with probability P. One variable being one is infected. And then we can compute the cost. Here we have a formula that one slide ahead. So this is the cost, and the argument is really the same thing we did with Dorfman. The first one, this one is just accounts for the first test, which we always perform. So if this test comes up positive, which happens with, so this is this parenthesis here, this expression in the parenthesis here is telling us that the first stage came up positive. And if this happens, if this is equal to 1, this we'll see is equal to 1 or 0. So if this is a 1, then we have to move on to the second stage, and such as here. And in the second stage, we will test M1 over M2 tests. In this case, M1 was equal to 9, M2 was equal to 3. So in the second stage, we have to do 3 tests. And this is the number that will appear here. How many tests we perform in the second stage, provided the first test came up positive. So Phi0 here is just a Bernoulli random variable, and we want this to be 1 when this first test was positive. And you can see from there, just this is takes a little bit of accounting. You do all this accounting, and you get that Phi0 is a Bernoulli now Q to the M1, because this will be 1. This variable will be let me see if this is correct. Okay, let's assume it is correct. Phi0, we want it to be 0 if there's somebody who is infected, and we want it to be 1 when not. Okay, it's correct. So sorry, Phi0 will be 1 when all the individuals in the initial pool were healthy. So that's 1 minus P to the M1. And we do the same with then we repeat this argument to get the second expression. Let me know, not go over it because I will mess it up. So you do the same, and you add 1 here. We're adding 1 from I equal 1 to 3 because that comes from considering this pool, and this pool. This is what the summation is over. And in the end, you get this expression here. You can check it. So, okay, so this is a random expression. It depends on how the values of the variables. But if you take expectations here and you divide by M1, so if you divide by M1, you will get a 1/9, M1 was 9. Then this M1 will cancel out with the denominator, and we get from here we'll get a 1 over M2, which was 3. And then here, when we take the expectation here, we'll have a 3 coming up from the sum, and then we'll have a 3 coming up from here, which will go away. And altogether, we will get this expression here. M3 was equal to 1. So we get that the cost is this. And now if we want to generalize it to just the same idea applied to the general case, we get this expression here in the box. Okay, so we have a very clear expression for the cost, whatever the choice of number of stages and the choice of the pool sizes. The problem is optimizing this expression. So now comes the game is to choose K. Your input is P, and what you want to do is choose K and choose the vector M of pool sizes so that you minimize this. So that would be what we want to do. And we have in mind that we know what happens with binary splitting. In binary splitting, we had the cost was of order P log 2P. So we would like to have something of that order so that it's comparable to binary splitting. Okay, so this is the outcome. Turns out that we managed, it took us quite a while, but we managed to reduce the possibilities to 4 possible strategies that are here enumerated. The first observation is that if your probability of being infected is above 0.30, so let's say 30% of the population, the infection probability is around 30%, a little bit below 30%, then the best, with this type of strategies, the best thing is to test everyone individually. Otherwise, you'd be wasting a lot of tests because a lot of first tests will come up positive, and then you will have to move to the second stage and test everyone. And this is a waste. You will have wasted your first test. On the other hand, if P is below this probability, so for relatively small P's, then we can find the value of K, which is more or less log 3 of P. Same idea as for binary splitting. Why log 3? Well, you will see next, because here we're at each stage we're splitting mostly in 3, like in the example, in 3 subpools. So that's why the log 3. And then one of the following is the optimal strategy. And the first one is like the one we considered in the example, in the last example, where we start with 3 to the K, and then we keep on dividing by 3 until the last stage when we get to individual testing. Then there are a second type of strategies that are actually sometimes the best, where you take a slightly larger first pool, 4 times 3 to the K minus 1, and then from them on you keep on dividing by 3. So there's a slight difference at the first stage where you take a slightly larger first pool. And then there are these M23 and M24. M23 is the same thing as M33, except that the last pool, instead of being equal to 3, is equal to 2. And M24 is sort of a combination of the previous ones. So we start with a pool of size 4 times 3 to the K minus 1 times 2, and then you keep dividing by, first you divide by 4, and then you keep dividing by 3. And your last pool is of size 2, and then individual testing. But in fact, we conjecture that 3 and 4, we couldn't rule it out rigorously, but we think that 3 and 4 do not actually occur, that actually the best strategies, optimal strategies, are of type 1 and 2, only that we couldn't rigorously prove it. We got help to do some numerical computations, and numerically it was corroborated up to P larger than 2 to the minus 51. 2 to the 51 is larger than the population of the world. So for infections, this would be completely irrelevant because there would not be anybody infected with this, with an infection probability less than P to the minus 51, but we couldn't really prove it rigorously. So more importantly, so for now I will concentrate on the first 2 strategies because we think that the other ones are irrelevant really. But what is interesting though is that this threshold we found, the 1 minus P, the 0.3066 on the first line, well, this was already observed because this was already observed by Dorfman, this was known. And moreover, actually it was proved in 1960 that if your probability of infection is actually larger than 0.382, and the precise expression is this, then simply there is no test that will do better than individual testing. All tests will be worse than individual testing. So if your probability of infection is kind of high, around 0.4, then it's best to test individually because everything else you will be wasting resources. And then recently, Vasilis Papanikolaou proposed another strategy which does better than individual testing for P below this bound. So really, this is the threshold. Above this, it's best to do individual testing. Below this, there are options that will be cheaper than individual testing. And here I have a picture, just to have a graphic idea of what's going on. So what we're looking at here on the x-axis, we have the probabilities. Notice that this vertical line is precisely at this threshold value of 0.30 something. So that's where we stop to applying one of these testing strategies, and we'll move on to individual testing. And then there is on the y-axis, what we're looking at the costs. So all these lines are associated to different testing strategies of the kind we are considering, nested testing strategies, the ones we are proposing now. And you see that when you go to when you stand on the threshold, you see that the horizontal line at 1 and this strategy overlap. They take the same value, and because at this precise probability of infection, the cost is the same. If you go above, then it's cheaper to do just individual testing. If you go below that, then you have options, and this is what we described here. And so, as I said, each line is associated to a strategy, and we want to take the one which is the cheapest. So the one we're looking at the lower bar, the one which is the minimum one. Okay. So let me see if I can. Okay. So maybe you can see better here what's going on. So the first thing we see, so we're moving from right to left. Okay. So we start at 0.30 something, and we see that the minimum is achieved at this dotted line. And this dotted line is associated to this strategy, which starts with this dot one, with the first initial pool of size 3, and then individual testing on the second stage. And this keeps on going until more or less here. Let me see. Right? And then it's not very, I mean, if I try to enlarge that, I don't think it improves. The resolution is not good enough. But then at that point, we move to this strategy here, where it's one of the ones with the 4s. So what happens is that the first pool is larger than 3, it's 4, and then second stage, you do individual testing. And that is a minimum for a very short while, after which you move on to this is the solid black line, then you move on to the dotted blue line, which is this strategy here. And now what we did is we add one extra stage. So we move on to the strategy where you start with 9, then you move on to 3. So first stage is pool size 9, the one in the example. Then you have 3 pools of size 3, and then at the last stage, you have this individual testing. All right. And you keep moving on, so you are decreasing P. The idea here is that this is intuitively clear. The smallest the P, so you can take a larger initial pool because you can put more people together because it will be harder to find someone who is infected. So if you take a larger pool, you could save more, you could increase your savings. So when you take P right around, I can't even see it, but it's somewhere around here, it seems, then we have a little bit of time in the p-axis where it's better to take the solid blue line, which is associated to, instead of starting with 9, then 3, then 1, going 12, then 3, then 1. So we take this 4 times 3, in this case, in the first pool strategy. And then we keep going. And so this keeps repeating. And here. So this is what really happens. We conclude this. The strategies, as you move towards, as you decrease in P, you move from right to left. What you see is that you have these transitions. You have a strategy of this type. Then as P becomes smaller, you just make the first pool larger, but you keep the number of stages fixed at K. And then eventually you go below a threshold at which it's best to add on a new stage, move on and have a new stage. And so you start with 3 to the K plus 1, then 3 to the Ks, and you keep decreasing. And so you have this kind of structure here. It's interesting. Here there's a log-log picture telling us where each of them holds, but this is, keep in mind, this is log-log, because this segment where it's best to take these strategies having a first pool, which is a multiple of 4, they really happen at very short intervals that we can actually describe precisely. Okay, so let me just tell you, the proof of the theorem is kind of, it's elementary. We get it by following very carefully a series of lemmas, but it's elementary, but lengthy. So essentially, I will just describe 3 lemmas. So that give our result, that give us our result. The first one is that the ratio between the first pool and the second one has to be 3 or 4, as we already understood that. The second one is that these ratios actually have to be non-increasing. So this tells us that, for instance, if our first ratio was 3, then when we go from the second to the third, the ratio can be 3 or 2, or 1 if we have a short, if K is small enough and the next step is just 1. Okay, so No, not one, sorry, I'm messing up. So if the first one was 3, let's go over again. If the first one is 3, then when you go from the second to the third, you can only have 3 or 2. Okay, and that's the first thing, and so on and so forth. So this restricts the strategies a lot. And finally, the third lemma tells us that there cannot be actually 2 consecutive ratios which are equal to 2 or 4. So now if you apply all this, it tells us that if you start with so you can start with the first ratio, which is 3 or 4. If the first ratio is 4, you know that the next one cannot be 4. It will be 3 or 2, because there cannot be 2 consecutive 4s. And so this forces you, actually, when you apply all this together, it forces you to, you could start with a 4 in the first ratio, you could end up with a 2 in the last ratio, but anything in between has to be equal to 3. And then, depending on the number of stages you're considering, this forces one of the strategies we had before. And then, of course, you have to determine what's the value of k that's optimal for your choice of p. And so this is one of our most, this is our main result, the one I described before. But in practical applications, actually, I mean, if you have a lab and you want to implement this, you like to have something very clear cut that tells you what to do, not have to start for each p that you have to estimate, start wondering. So in fact, as a take-home message, our suggestion was ignore all strategies, but the most common one, which is the one where we take k equal to precisely this value here. And this value here, when you work it out, is essentially to first order just log 3 1 over p. Okay, so this is the or log 3p absolute value. So this is the number of stages, and it's deterministic. And then we show that whatever your optimal strategy and the cost of your optimal strategy here, this is the cost of the optimal strategy. If you want to compare it with the strategy consisting of taking this value for k and then taking the pool sizes, which are power of 3, associated to this particular value of k, then the cost will not be too far away. Actually, it will be of order, plus corrections, 0.013 p log p. And remember that this is, sorry, this is the order at which we are interested in working. This was the order of the cost, the cost order for binary splitting and for the optimal bound we had before. So the difference is small in this order. And moreover, we can bound the cost by this value here. So we will see at the end how this compares with binary splitting, but at least we're working in the same order. It's slightly worse than, it's quite worse. I mean, this is like, I think, 278 or something like this. And so it's larger than what we get by applying binary splitting, but not too much. And the advantage is that this is a very simple method. Now you start with a pool and then you keep on dividing by 3, and you know in how many stages when you perform all this in parallel. After so many stages, or the log 3 1 over p, you will have determined the status of all the individuals. So it's very easy to implement. And then we in another paper, now same people plus Hugo Mencela, who's a chemist working here in the Universidad Nacional de Rosario, we studied more like practical aspects of the implementation, such as what happens if you want to estimate p, for instance. If you don't know p a priori, how do you go about estimating p and performing tests simultaneously so as to be as economical as possible? Also, there are other issues. For instance, what happens with how this algorithm flows. For instance, one concern you might have is that, so you're running all this in parallel, and you have a lab, so you have limited resources, limited processing capability. So one thing that could happen here is that, so we're taking one pool and splitting it in 3. Imagine that our first pool tests positive and things keep testing positive at each stage. So we're multiplying by 3 the number of tests, necessary tests at each stage, and this blows up. I mean, eventually you have too many tests that you need to run in parallel, so you have to leave some aside, and so it will become harder to organize this, to manage the situation. So the idea is that this doesn't happen because many pools will test negative. It's true that every pool that tests positive will be split in 3, but on the other hand, pools that test negative are removed. So hopefully these things balance out. And in fact, we can study these because we're able to compute the variance of the variables we're studying. So we can check how this flows. So that's what we did there. And maybe I'll skip this slide. Just say a few words about one last proposal for pool testing, which is non-adaptive methods. I just want to mention this briefly. So non-adaptive methods are the ones where you decide, I mean, what we discussed before was probabilistic, we discussed this already, but it was also adaptive. Adaptive means that at each stage you use the information acquired in the previous stages. So we decided what to test in each stage, decided depending on what had happened in the previous stage. Non-adaptive methods are the ones where you decide a priori before you take the samples, you devise your method and you know how you're going to distribute your samples, how you're going to form the pools, and so everything is pre-decided. And it turns out that this is not so good for probabilistic testing. If you're really interested in determining the status of each individual and you want to go fully non-adaptive and plan everything ahead, then the best you can do actually is just test individually, and that's it. But on the other hand, you can compromise and you can say, well, okay, I like non-adaptive methods because they're easier to organize ahead. And then, okay, at the last stage, everything that I have not been able to determine, all those variables whose status has not been determined, we can test them on a second stage and be done. And so this actually, this was implemented, I should say. This was implemented in Argentina, in La Plata, which is the capital city of the state of Buenos Aires, by a group of scientists led by Daniela Hosford, who works in the Universidad Nacional de La Plata, and Roberto Echenique, who's also at the Universidad de Buenos Aires. And here's a link to how they implemented and the results they got. And now for rigorous results on the two-stage algorithm, I'll just mention this one by Mark Mezzard and Cristina Toninelli from 2011. And what they propose, let me just say quite briefly what they do. So the idea here is, this is different from what we were doing before in that if you remember what we did, either binary splitting or a nested procedure, at each stage, every sample was tested and was tested in just one pool. Whereas here, what we propose is that a sample can be tested. Suppose you have a blood sample, so you can split it up in many sub-samples, and then you can test the same sample in several tests simultaneously. And hopefully you'll get some information from this. So you can organize your information, think that you have a matrix, which is called the connectivity matrix. So you decide that you're going to form M pools, and you can think that each pool, you can think of it as a string of ones and zeros, C1A and 2C and A, and is the size of the population, and these are just one if the ith variable is in this pool, zero otherwise. And this gives you a matrix having, say, n rows and m columns, and the entry in the position IA is just one if the ith sample is considered in the 8th pool. So this is the connectivity matrix. Or alternatively, you could think that you have a graph, such as here, where you have the samples on one side and the pools on the other, and then you link them if this sample belongs to this pool. It's another way of thinking about it, of keeping this information. Then you test them, and you will say that the test is equal to 1 if it turns out that there was some infected sample in the particular pool, 0 otherwise, just as before. And when you finish this, you will have, say this is the first stage. And when you're done, you have you can organize your variables in 3 groups. On the one hand, you have what we call the identified zeros. So are those variables that by the information you will obtain by this pooling and testing procedure, you can certify that these samples are not are zeros, are not infected. And that would happen if it turns out that one of the pools, at least one of the pools, these variables belong to tested negative. Then you can clear them out. So these are the identified zeros. Once you know your identified zeros, then you have your identified ones. How could you have so the ones that you can sort of the variables that you can say with information you already processed that they're infected. And this would happen if a variable is in a pool which tested positive, and you know that all the variables, the other variables in the pool were identified zeros. So you first get your identified zeros, and then if you can tell that everyone else in this pool was negative, then and the pool tested positive, then you immediately conclude that this particular variable was positive. So these are the identified ones, and then there's everybody else, the unidentified variables. And as I said, on a second stage, you test these unidentified variables, and that's the test. And the goal here is to control the cost. And here's the cost. So first of all, you have the M pools you test on the first stage. So this is deterministic. You have to test them. And then on the second stage, you test the unidentified variables. So you have two types of unidentified variables. You have those which are the unidentified zeros. So these are these. And the absolute value means the number of the cardinality of the set. And then V here is the set of unidentified ones. And in order to compute the cost, you have to take the expectation. And well, you take the expectation for the V's, which are the ones. Actually, this is negligible because you notice that the set of unidentified ones is contained in the set of ones, and the set of ones has order NP because this is actually equal to NP because these are the non-random variables. So an NP is, we're working in the order P log 2P, and we're assuming that P goes to 0. So if P goes to 0, this is small, this is negligible compared to P log P, NP log 2P, so we can just neglect this. And then the game is in minimizing the number of unidentified zeros. And okay, I don't think I have time for this, but you can read it here. There's some magic in it, and the magic is that you can write you can get the number, you can quantify this by writing it like this. This is an inequality, this is an identity, and I describe here. And the nice thing is this is an algebraic expression that will let you compute expectations. And then you have to compute the probability of the expectation of some product. And turns out that in some good situations, this just factors out, they're independent. And when they're independent, you can compute them explicitly. And you just get a theorem that tells you that the cost, when you normalize by considering the total population, will be this quantity here. There are some assumptions here. Again, they had to play around and optimize. And these are the optimal parameters. So the number of tests on the first stage is given by this quantity. They have to fix the degree. If you think in terms of the graph, you have to fix the degree of each sample, and you have to fix the degree of each pool to these quantities here. And when you do all these properly, you see that this is not empty. So you have graphs like this that will allow you to do this. And then you will end up having this cost. So now, as a last slide, let's compare them. So there are three proposals here. The first one is binary splitting. This is best in terms of cost, but as we said, there's a random number of stages, and this is harder to manage. The two-stage algorithm has slightly worse cost. It's 208. It's the last one we discussed. But on the other hand, it's just 2 stages. That's a big plus. And the downside is that it requires running this order. This was the order of the number of pools we had to consider on the first stage, and this is quite large. It's order n p log p. And finally, we have nested pooling, still on the same scale, but slightly worse than two-stage algorithm, really. But the advantages, there are 2 advantages. Compared to binary splitting, there's a fixed number of stages, precisely this, which is like log p, really, on the 1 on p. And the other thing is, if you compare it to the 2-stage algorithm, the number of parallel tests now is smaller, is order NP. So in this sense, you need less resources. to be able to run it in parallel. And I think this I should have stopped 5 minutes ago, so I need to stop here. Just one last slide. If you want to play, here there's a web page where you can play, enter your value of P and some other parameters, and it will tell you what's the best strategy. This web page is being maintained by Francisco Sobral from the Universidad de Maringa. So you're welcome to play around. I like advertising this. I have nothing to do with it really, with the development of this web page, and I think it's really nice because it lets you really speedily compute what you should do if you want to apply this. So, and then here are some references. So I'll stop at this point.
Thank you, Inés. It was a really interesting talk. So now we are going to open to questions. So if you have a question, you can mention it on the chat or just write down your question and I can read to Inés. So I have a question about the last procedure that you show about construct this graph, because we have a kind of two-stage procedure. And then I was wondering if I don't know, when n is large, you can construct the graph, test, and then you have individuals that you want to test again. Then instead of doing the individual test, you build a graph again, the pools, and make the test. I don't know if it makes sense or the cost will be expensive in this way.
Yeah, I mean, this was not considered in the paper by Cristina and Mezar, by Mezar and Toninelli, but you could do such things. I mean, afterwards, you can do several things. You could combine, actually. You could take this and then do binary splitting once you've reduced your number of the size of your sample to something more controllable. You could apply another algorithm. You can combine them any way you want. And of course, you can also consider other types of tests where you do non-adaptive in several stages, of course, yes.
Ah, I see, okay.
Yeah. This was not included, but you could do such a thing.
Or for example, construct the graph, detect the individuals that you want to test again, and then do this kind of nested spool as you show, and then.
Yes, absolutely.
I see, okay. Thank you, Inés. Let's see if we have more questions. If we don't have, I really want to ask something very, very quickly. So you show the optimal cost in slide 52. I was wondering what's Q, because I was a little lost. Is 1 minus P or?
Where was that? I mean, you mean?
Is slide 52? Yes, when you show the optimal cost. It's your theorem.
Yeah, maybe I should not do this.
Yeah.
I think it's here. No, you mean here.
No. Move forward. Yeah.
You mean this one? Okay.
I think it's the next one. Yeah. This one. So you have Q. No, Q, and I don't think my phone. You have
Q. Yeah.
Q is 1 minus P, or is a different? So
yeah,
K
here,
depends on P.
Yeah, you have to think that this is log 3, and then if you want, maybe it's better to write log of 3, that log is now natural log, and then you have log 1 minus P.
Okay, so when you talk about estimate P, it will help you to find K.
Yeah, I mean, here we're assuming that P is known, so you know what you're going to do. In practice, you have some estimate, you have an idea of what you're going to do. More or less, you know what P should be, but you don't know it precisely, because the idea is that maybe you have different populations. For instance, population, if you're testing people working, staff working in a hospital, it should be different from something else. I mean, it could depend on the local population. So the idea, this is something we discussed in the other paper. The idea here is actually you should be very conservative. Be conservative. Take your think that P is as large as possible. So start with something that could turn out to be the best way is take your initial pool associated to the largest value you think could apply to P.
Okay.
So this means you're starting with a quite small initial pool, because pools go up, initial pools become larger as you decrease P. So, and then you investigate these, and on the second stage, you will have gathered information about P. So you can readjust, tweak with your method and apply it in a more efficient way. So you need to do something like this if you want to estimate P and test at the same time. So as not to waste time.
I see. Okay, now I understand. Okay, so thank you again, Inés, for accepting our invitation. It was a really nice talk. I hope to see you soon, maybe in Brazil or in Argentina, I don't know.
Yeah, let's hope so.
So thank you. See you.
Thank you. Bye.
Bye-bye. So I'm going to invite Michelle to introduce our next speaker.
Márcio, you there?
I am here, yes.
Oh, great. Want to try your slides?
Give me 1 second. Can you see my slides?
Yes.
Yeah. Okay, great.
Yes. Excellent.
Let's see.
Okay.
The chat box.
Okay. Going on with the workshop. Now we have an online conference entitled Shrinkage Priors for Single Cell Experiments. This is the work by Márcio Diniz. Márcio Diniz is a Brazilian friend. He is a statistician. His bachelor was in UNICAMP, and his PhD was also in statistics at University of São Paulo. Now Márcio is assistant professor at the Cedars-Sinai in Los Angeles, California. Among other things, his interests include adaptive trials, such as those found in phase 1 trials, basket trials, and seamless phase 2-3 trials under a Bayesian framework. Márcio, thank you for accepting to giving us this talk today, and please feel free to start when you're ready.
Yeah, thank you, Michelle, for the kind introduction. It's always a pleasure to be among friends. So today I'm going to talk about shrinkage priors for single-cell RNA-seq experiments. This is ongoing work with Nathan Ng and Simon Knott. They are both bioinformaticians here at Cedars-Sinai, and they approached me December last year with this problem. So this work is still in early stage, but it's an interesting problem, and I thought it would be nice to share with you guys, and also maybe you can give me good suggestions how to solve it. So let's start with the first introduction. I was not very familiar with RNA-seq last year, so I had to study a little bit, and this is my understanding so far. So living organisms are formed by billions of cells, and each cell is driven by their genetic code, and that is contained in their nucleus, as we learned in high school biology, right? So in the nucleus of cells, we have the DNA molecules, and these DNA molecules, they are packed into the chromosomes. So the regions of chromosomes, each of them has some specific functions, and that's what we call genes, right? So biologists and bioinformaticians, they are interested in measure which genes are active or not. So a gene is active when they are transcribed by RNA molecules into proteins. So therefore, to measure the activity of a gene, what we can do is measure the number of RNA molecules associated with that gene. this is a very simplification of this whole process. But the idea is, if you have a large number of reads, means that the gene is very active. If you have a small number of reads or 0 reads, means that that gene is not active in that process. So RNA-seq was a technology introduced in 2006, early 2000s, that allowed the quantification of gene expression without any prior knowledge. Before that, they had something that they call microRNA array, which allowed us to do the same, but we had to predefine which genes we want to quantify. So RNA-seq was a huge change in the paradigm for in biology, in basic science, because now you could just quantify and see all the genes that are active in a given sample. Of course, that require a lot of bioinformatics analysis that I'm not going to enter in details here, such alignment, maybe QC, and normalization. So once they apply all these steps, then we can apply more traditional statistical tools that we see in a PhD in statistics, for example, or a master's degree in statistics. And then recently, there was the introduction of what we call single-cell RNA-seq. So what's the difference? So in the bulk RNA-seq, we are quantify the average of expression for a gene across a large population of cells. So basically, we are counting the number of reads for all the cells that are in a sample. In single RNA-seq, we are able to count the number of reads for each cell individually. So in this case, then we have the distribution of expression levels for a population of cell. We don't have anymore just a point estimate. So we have this whole distribution. And then what happens is that this technology was introduced in 2009 and has been more accessible since 2014 and has been even more popular now. Requires some additional steps, and a very common final analysis for RNA-seq for single cell is that in the end, we use some sort of clustering method to cluster those cells based on their genomic gene expression in clusters. So each cell now has a whole gene expression, and then we can cluster that. And you might have seen familiar map plots like that. And then here we are using UMAP, and we can define those different clusters of cells based on gene expression, right? So this is just a very quick and short introduction for RNA-seq for single cell. So once you have these clusters, that's what Simon Knott and Nathan Ng worked on. Then they want to give a step further. So they asked, well, changes in gene expression can be markers of treatment response. In particular, for single-cell RNA-seq, change in gene expression implies into change of the composition of those clusters. Therefore, this compositional vector indicating the proportion of cells in each cluster can be used as a marker of treatment response. So let me just go back here to the plot. So what happens is that for each patient, we have a plot like this one here. And each dot here, it's a cell. What happens is we can count the number of cells in each of the clusters. And at the end of the day, we have the proportion of cells in each of those clusters. What we hope is that once there is some sort of change in the biological process, such as treatment response, this proportion of cells in each of those clusters also are going to change, right? And what they are wondering is if we could use that as an indicator that, well, are the patients responding to the treatment or not? The first challenge was that a single cell, when you have a vector of proportions, we call this a compositional data. So let's say that we have the compositional vector here, x1, which is the proportion of cells in the cluster 1, xd, which is the proportion of cells in the cluster D. Adding up all these proportions, you have a constraint that you should add up called 1, right? We can't apply standard statistical tools in composition of data because the unit sum constraint generates a correlation structure among those components. So back in the 80s, Atkinson was aware of that, and he wrote a whole book about composition of data. And at that point in time, he proposed to transform the observed variables in the simplex 01 to the real space, applying this transformation called additive log ratio, which would take each proportion, divide by a given proportion of reference, and then apply the logarithm. Once you do this transformation, your data could be now modeled by a normal distribution, for example, a normal multivariate distribution. And then over the years, they had other proposals such as the center log ratio transformation, and also the isometric log ratio transformation that was more complicated to express mathematically here. But the idea is that you're going to break the proportions in two sets, and then you pick the big set and you break again, and so on, as a stick break procedure. Right. Okay, so in order to analyze any single cell data, we have to use tools that take into account the compositional nature of our data. And then the following questions we had is, how do we identify which cell types are associated with response, or how do we identify how relevant are the cell types? Because we can say that each cluster of cell is a different cell type, and we want to say, hey, which ones are associated with treatment response, and which ones are important, and which ones we can maybe ignore. They have a few approaches available in the literature, and this is a very hot subject right now in bioinformatics. So the last publication here was 2021, and they used univariate tests based on the Poisson regression. They use Wilcoxon tests after this additive log ratio transformation, and they recently they borrowed some ideas from from microbiome analysis, and they start using this Bayesian Dirichlet multinomial regression model with spike and slide priors. But in all these three approaches here, including this last one here, the single cell RNA-seq are the response variable instead of the predictors. And the problem that I had was slightly different, because my response variable is binary. I have treatment response, yes or no, and my predictors are the single cell data. So the question we want to answer is, how can we predict the treatment response based on single cell data? And how can we adjust for other clinical variables? Because even if I find differences between responders and non-responders in single cell, that might be for other reasons beyond the changes in the genetic expression, such as stage or of the disease, such as age, sex, and so on, right? So that's when I start studying a little bit more about sparsity under a Bayesian framework. So sparsity in the Bayesian framework, it's more well known as, for example, lasso or elastic net, and all those different penalization methods that we have in the frequentist framework. In Bayesian, there is two main approaches that generate sparse solutions. One is called spike and slab, and the other one is called shrinkage priors. So spike and slabs is basically we give a discrete mass of probability for beta equals 0, and then we also give a continuous distribution for all the other values of beta. That's our regression coefficients. The problem is that we are doing this mixture, and then there is a computational effort can be very large because we start thinking about all the possible combinations. So it's interesting and it's applicable, but we would like to do something slightly different here. And we opted to pursue shrinkage priors because they are more attractive computationally. The problem with the shrinkage priors, and we are going to see these problems in our application here, is that we don't have posterior probability mass on beta equals 0, right? Because we are treating beta as a absolute continuous variable. So the probability at a specific point is 0. So different from the classical solutions such as lasso that we have coefficients that are equal 0, shrinkage priors are never going to give us truly sparse solutions. And that might be a limitation. But let's give a try and see, and we decided to give a try and see what we'd get from that. So this is the model that we are trying to adjust. It's a logistic model. Our response variable here, treatment response, yes or no. It's binary. We are modeling by a Bernoulli distribution. And then the logit of the probability of treatment response is modeled as beta 0 plus this standardized and scaled covariates here and a vector of parameters, right? So this standardized design matrix here, it's our proportions of single cell RNA-seq, but after the transformation. And in particular, we chose to use the center log ratio. That was the second transformation that was proposed for compositional data here. And we chose that because for all the other transformations, we always lose one dimension. And they did not want to lose or choose a reference. So we decided to use the center log ratio transformation. And then I did some literature review, and I found these different shrinkage priors. If we just use normal distribution for the parameters beta, we get a solution that is very similar or equivalent to the rigid regression in the frequentist framework. If we choose a prior for the beta as double exponential, then we get a solution that would be similar or equivalent to lasso. And we also have a equivalence to ElasticNet. And recently, they have proposed this prior called horseshoe, and then a variant of horseshoe, it's called regularized horseshoe. And also they have this another new proposal, logit normal. So we decided to explore all these proposals here. All those distributions, they can be written as a normal mixture scale. So the simplest one, as I told you, is just give a normal distribution prior for each beta, right? So there is no, and then we choose a hierarchical framework such that tau, which is the variance of the prior distribution, follows a half Cauchy distribution with a parameter tau 0 here. And then for lasso, for example, or the double exponential, we are able to write the double exponential distribution as a normal scale mixture, such that the lambdas, which are the weights of the mixture, follows a Hailey distribution. And again, tau is a half Cauchy. So ElasticNet is a more complicated scale mixture. In this case here, we have tau 2 and tau 1, and lambda follows a gamma distribution. We have horseshoe. In this case here, lambda and tau, they both follow half Cauchy distributions. In particular, the weights are half Cauchy 0 and 1. And then we have the regularized horseshoe, which is a little bit more complicated. And they propose the regularized horseshoe because what happens is that for very large values of beta, the horseshoe doesn't work very well. So the idea is that for small values of beta, the regularized horseshoe would be very similar to the original horseshoe. But for very large values of beta, then beta would follow a normal distribution with scale c square. And in particular, if we give it a distribution for c square, in this case inverse gamma, then the beta j's here, the prior distribution becomes a t student, and then we can calibrate these two students accordingly, right? And again, we have, excuse me, we have tau as a half Cauchy and the hyperparameter tau 0 that we are going to calibrate. And finally, we have the logit normal here, where our weights follow is the logit of a normal distribution. So those priors, they look all very, very similar, right? But they can be differentiated. And the way we do that is based on the global shrinkage. So if our case is that we have Bernoulli distributions, but for normal distributed variables, we can write the average of the posterior distribution for the betas, such as these. And based on where beta hat is the maximum likelihood estimator. And we can say that kappa j is the shrinkage factor. And kappa j actually has a formula such that if you see kappa going to 0, that means that the posterior distribution beta j bar here is going to be equal to the maximal likelihood estimation. So there is no shrinkage. If kappa goes to 1, so there is more shrinkage, and the estimate of the estimated given by the posterior distribution is going to be a shrunk version of the maximal likelihood estimator. Right, and we can see that kappa is a function of the sample size, of the variability of my data, of tau, which is the what we call global shrinkage, and also of the weights, which is the lambda. So these two guys here in 2017, they suggest to calibrate the prior distribution based on the parameter tau 0. Remember that tau follows a half Cauchy distribution with parameter 0, tau 0, based on the effective number of non-zero regression coefficients, which is basically the sum of 1 minus those shrinkage factors, kappa, here. And they also proposed, so these results that I just showed are for the linear normal distribution, and they also propose to adapt here when we have Bernoulli response variables. So now we can show how those priors are different somehow here. So imagine we have 10 cell clusters and we have different proportions of response, 10%, 30%, 50% here. You can see that, for example, the regularized horseshoe, which is the blue, they give more prior distribution for a smaller number of clusters. Then follows the logit normal prior distribution, and then we have the double exponential, which is this variation of green here, and then we have the horseshoe, which is actual green, and finally the rigid regression gives a very large probability for all the clusters, which is expected. So that's the idea of rigid regression. We should rank, but we should rank equally all the parameters. If we have 20 clusters, then we can see a little bit more separation between the priors, right? So the question is, well, would these priors lead us to different results? Also, as I mentioned, there is a challenge here because how can we do variable selection for a shrinkage priors? Because those penalization methods, they do not shrink the coefficients exactly to 0. In this case, there is not really an optimal solution, but the main approach is to use high probability density intervals. And this group of researchers here, they chose different levels for different priors, but they did not study the legit normal shrinkage prior. I think I just need to go quickly here. So we generate our data following one of those papers proposing different methods for single cell data. And the idea here is that we are generating our cell counts using the normal distribution, but the probabilities for each cluster, they are defined using what we call a softmax transformation over a normal d-variate. And then we can play this normal d-variate with a correlation structure, although in particular the paper that I based this generation process did not add a correlation structure, and we just followed their reference. So we did the simulation study where we generated 10,000 cells with different number of clusters, 10, 20, for different sample size. And this is a large, 100 samples is a very large sample size. 20 and 40 is a more reasonable sample size. That's our example. And we fixed our coefficients here. A few of them are different from 0, and the remaining of them are equals 0. And we try to study some operating characteristics. So the first set of operating characteristics that we started to study was, well, to understand the biological process. And in this case, here we have the false positive rate, the false negative rate, and also the correlation between the true and the estimated beta regression coefficients here. Right, and then finally, we also want to talk about, well, can we use RNA-seq as a treatment response marker? So in this case, we are talking about prediction, not inference. And then we have AUC and Breyer score here. So we run the simulations and we showed that for all those different priors here, we are able to control the false positive error positive rates here. In particular, the horseshoe and the regularized horseshoe does this much better than anything else. What else? So, but we don't control, as I told, the false, all this process of choosing variables based on the high probability density, high probability density intervals is a very conservative method. And in the end, even though we are controlling the false positive rate, we have a very high false negative rate. And I'm jumping my slides here. So, and as you can see here, we have, for example, for 10 clusters, we have a false negative rate of around 8%, and for 20 clusters, we have this less, but that's because we have less chance to do a false negative in this case, right? So that's not necessarily very ideal. But interesting, when we calculate the average spiral correlation between our estimates and our true values, as the sample size increases, this correlation increases, and that indicates that even though we are not able to point which cell type clusters are like important or not, because false negative rate is high, we are still at least able to order the importance of the cell clusters here. And finally, we're going to look at the AUC here, and we see also that the AUC is good and more or less the same for all different priors here, except the legit normal prior that has a lower AUC in some cases here. Oh, I don't have the Breyer score. So also there is one plot that is missing here, which would be the Breyer score, and the results are also equivalent among the priors. So finally, we have our application. So we chose in the end the regularized horseshoe because we are able to control a little bit more the the false positive rate than anything else. And also the regularized horseshoe is more efficient computationally for us. So in our application here, we have a phase 2 study, and we have this preoperative combination of this treatment, pembrolizumab, and also radiation therapy in 35 patients, and they have all breast cancer. So, and out of these 35 patients, 24 had complete response, and we want to say, hey, can we use the RNA-seq to predict this treatment response? If yes, which cell types are important? So this is more or less our data. So here we can see the proportion of cells for each of the clusters we have, and the blue are the response and the red are the non-response. So in the end, this is the posterior estimate of the regression coefficients, such that the out interval, which is the thin line, is the 95% HPD, and the inner, the sticker line, is the 70% HPD. So we can see among all of them that most of the coefficients were shrunk to 0, except the cluster 11. But even the cluster 11, the 95% HPD includes 0. So if we consider 95% HPD as our variable selection method, all these coefficients are 0, and we are being overly conservative. If we follow one of those references that I mentioned that suggests 70% HPD, then we get the cluster 11 here. If we look the shrinkage factors for each of the clusters, we can see, remember that 1 is high shrinkage and 0 is no shrinkage. So we can see that the cluster 11, which is this shade of blue here, is the only one that doesn't suffer shrinkage in comparison with all the other ones. So this is the prior distribution here doesn't change very much for all the other clusters. So we can say that, well, cluster 11 probably it's the most interesting one here and the most predictor of our treatment response. We can see also the posterior distribution for the effective number of non-zero regression coefficients. And this posterior gives us 10 clusters, which seems overly optimistic, because if you go back here, maybe we have 10 different clusters that are important, but probably not. So that's probably not necessarily a good idea here. So just a few conclusions is, in this case here, we can apply those shrinkage priors for a single cell RNA-seq. The main shrinkage priors don't have large differences in the operating characteristics. And finally, we cannot control explicitly the false positive rate and the false negative rate. And that's a problem, right, to understand which effects are biologically relevant. So we are working on this right now and trying to find better approach than the high probability density intervals. And among all those priors, we suggest the regularized horseshoe or the horseshoe, because they show better control of the false positive rate. And at least those shrinkage priors allows us to order which cell types are important, and they are all useful to build predictive models that eventually will be used as treatment response biomarkers. Our future work that I'm accepting ideas and collaborations, how can we choose and how can we define relevant cell types, why we control the false positive rate. And that's all. Hopefully I did not go over my time very much.
Thank you, Marcio. Thanks for bringing this interesting problem and for the nice talk. Before I open to questions, let me be selfish and ask one question myself. Marcio, as you know, I am not an expert in this topic, but it sounds very interesting. And just to understand it better, you have compositional data because you have clusters. And these clusters are predefined. Is it?
So yeah, so what happens is the clusters, they are defined by data-driven methods. So they use different methods. So they are not necessarily predefined. They are defined based on the data.
Okay. And for when you select just a few coefficients, then you are reorganizing these clusters or not?
No, not really. So what I'm saying is that, well, for example, the cluster 6 is important. So then what I do is I go into the cluster 6 and see what's the expression of the cluster 6, right? So in the sense that the cluster 6 has gene A and gene B overly expressed, and so I don't reorganize. So basically what I'm doing is I'm using just as a proxy to identify which genes are important here. So that's what I'm going to do in the end, right? So I'm just going to, the structure of the cluster is going to keep the same, and they say like in cluster 6, gene A and B are most important. In cluster 7, gene C and D is most important. So that's we keep them fixed, yeah.
Okay, but they can be, for example, thrown to the trash, right? By the selection.
Yeah, exactly. So basically what I'm saying is that I don't care about cluster 7, 0, 2. I only care about cluster 6 and what's the proportion of this cluster 6 over the overall number here. So that's what I'm saying. How many cells I have here, consider all these other clusters, like over the total number of cells, right? So how big is this cluster 6 in comparison to the total? So that's, yeah.
I see. So, but let me understand just a little bit more.
Of course.
If you have so basically your covariates are compositional. But do you have any other variables? Any other?
Yeah. So in the end of the day, we did not get there yet. Is that we have other patients' characteristics that we also would like to include and see if those clusters are still important after we adjust for those clinical variables. So that's what led me to consider the shrinkage priors and use the compositional data as predictors. So yeah, we do have it. So for example, we have stage of the disease, we have age of the patient, treatments, previous treatments. Some patients, they could have gone through different treatments before, and so on.
That's my point. So what would be the impact of adding this covariates in the model? Would it change anything?
So what we do is, in a second, we not change in terms of approach. What we do is we would see if the single cell adds predictive power after we say those variables, because those variables, let's say stage of the cancer can predict we already know that stage of the cancer can predict treatment response. But then we would say, well, if I use single cell, can I get a better prediction in addition to stage of the disease, for example? So yeah, but we still apply the same approach, and we still be able to apply shrinkage priors to this covariates. So that would be okay. So we do the selection over all those covariates as well.
Yes, you are talking about something that I want to ask another question, if you don't mind. It's about the shrinkage priors. You show that when the kappa goes to 0, you have something. When it goes to 1, you have something, but it actually doesn't really can be 0 or 1.
Yes, yeah, exactly. So the coefficients will never be exactly called 0, because the distribution of the beta is a continuous distribution, so it will never be exactly 0, right?
So can you not, for example, to give us some kind of prior to the kappa, so that you can put a point of mass and 1 or 0 and have your selection?
So what happens is when we choose one of those priors here, we give indirectly a prior to the kappas. So that's why, for example, prior called horseshoe is called horseshoe because the kappa looks like a horseshoe. So we are doing that indirectly, but still it's a continuous distribution for the kappa, right?
I see. I see.
Yeah. So each of those priors is going to give a different distribution for kappa. And then, for example, I did not put here the plots, but for the the lasso, the prior for kappa is something like this. So they differ depending on the prior you give to the tau, which is kappa is a function of the tau. So we define depending on the tau and lambda, kappa has a different distribution. Like indirectly.
Okay. Thank you, Marcio. And I'm sorry, Diane. Diane asked that question and I was polite.
Let me answer. Let me answer, Diane.
Diane can turn her microphone and ask.
Are you listening? Yeah?
Yes.
Yeah. Hi, Marcio.
Hi, good to see you.
Hi, how are you? Nice to see you. Congratulations for the talk. It's a very interesting subject. Usually, I work with genetic data sets.
Oh,
and
cool.
I'm trying for the 2.5 in a year work with a single cell, but I don't have time to start it yet.
Okay, yeah, no.
And my question, I'm not sure if you observed this in your data set because you work with clusters, but when we are talking about single cells, usually we have a zero inflated data set.
That's true. That's true. No, you're correct. So what happens is, let me just, so they have already proposed that for microbiome data. So among those, I don't have that for single cell, but let's see here. So they usually have this, they have a Bayesian Dirichlet multinomial model with inflated zeros. But here in this, so, but the response is inflated with zeros. So this, because in this strategy here, the single cell is the response variable, right? So then you have all those inflated zeros. So when you are working with compositional data and is a covariate, then the usual strategy used for compositional data, which is not great, they have two. One of them is just to give a very small number and then to not let that composition say to be exactly equal to zero. And the other one is the other one is they do a strategy of imputation, but that's when we are using as a covariate. So if single cell is a response, then you have this option, which is the Dirichlet multinomial model with inflated zeros.
And you deal with this as a predictors, but in the other hand, if you have usually predictors that vary a very low variability, it's very hard to select the relevant variables.
Yeah, it is.
Did you notice that this problem or no?
Exactly. So then what was our solution was to standardize those variables. So we centered and we scaled them, because then we put all the predictors with mean 0 and standard deviation equal 1. And then once we do that, we get rid of this issue, right? We are not able to recover the same betas because the design matrices are different. So we are generating the design, we generate the data with the original scale, but when we are estimating the model, we are using the standardized data, but we are still able to do inference and at least estimate the order of the predictors and so on. But yeah, that's true.
Okay, thank you so much and congratulations.
Of course, yeah, my pleasure.
Well, let me see. Okay, we don't have more questions and no more time.
Okay.
So, Marcio, thank you so much for accepting and giving us this talk today.
Yeah, my pleasure. Okay, bye guys.
Bye-bye. Now we are going to the student talk. So we have Rodrigo Lassans to give his talk today.
Hello, everyone.
Hi, Rodrigo.
Let's see.
Wanna try to share your screen?
Sure, just a minute. Okay.
Okay, so we'll leave the questions to the end. Is that okay?
Sure, of course. Okay, so may I start?
Yes, wait. Now we have Rodrigo Lassans, and his talk will be about parametric pragmatic hypothesis testing. Okay, Rodrigo, thank you. You can start now.
Thank you, Michelle. Hello, everybody. Good afternoon. So my talk here will be about non-parametric pragmatic hypothesis testing. So in these 20-25 minutes, I'll try to break down each of these terms in a way that makes sense to what we're going to present. Okay, but first, I guess we should focus on the hypothesis testing part. So we're doing something that is, in a way, a proposal of change of the standard way we do hypothesis tests today. Okay, so we should first, of course, speak about what I'm calling here to be the crisis in hypothesis testing. All right. So when we talk about standard tests, we have a procedure that have garnered a considerable credibility in different scientific areas throughout time. And this is, of course, due to the fact that these procedures allows us to derive conclusions even when we have random effects on our data, right? And in general, they have been shown to be a trustworthy procedure to allow us to reject hypotheses, right? However, in recent years, we have seen a crisis in this credibility, so to say, and this has been due to some factors, but mainly we could blame maybe the replication crisis in areas such as psychology, when we have, a result that was significant, when we try to replicate it, we just fail to replicate it, it's not significant, so we're not sure what to trust, what not to trust. We have big data issues where it's become common for people to say, oh, if you have a big enough sample, you can practically reject anything you try to attempt, which I don't think it's exactly true, but still it reflects a thing that we are trying to bring here into the highlight, okay? And there is, of course, a misinterpretation of results when the test is designed to do something, but the researcher is trying to do something a little bit different, right? So let's wrap up the main issues we have with standard tests. The first one I'm calling unintuitive outcomes, right? Which is basically, well, as a researcher, you usually want to find out if you should reject a hypothesis, accept it, or remain in doubt about it, right? But when we do a standard test, we end up fusing these two states here into the state of do not reject, which we are used to do. So it is common practice in statistics to say you can reject, but you can never say you're accepting. You need to be more careful in order to talk about acceptance of a hypothesis. However, that usually is inside the group of things the researcher wants to know, right? The second issue, which I'm calling logical inconsistency, is something like, well, I could derive multiple testing and then have something like do not reject or accept the first hypothesis, accept the second hypothesis, but reject the third. Well, in this case, if A equals B and B equals C, this should lead us to believe that A equals C. But we have seen time and time again that standard tests can come to this kind of conclusion, which sometimes it is sort of confusing to the researchers. And the last part, which we will be focusing most on this presentation, is what I'm calling the rejection of irrelevant no hypothesis, which basically goes like this. Let's say you have a model, you estimate your parameter, and you have something quite close to 0. And for practical purposes, you could ignore that factor and say that is equal to 0, or that it can be disregarded. It's not relevant for your model, etc. But still, since that kind of information is not contained in the test you're deriving, you could come to the conclusion of rejecting the parameter to be equal to 0. And this is a sign that we're not into our test and information comes from a research area, and that it is important and somehow to put it in. Okay, so let's talk about the big questions which this presentation seeks to answer, right? First, how do we derive a test that is not affected by these issues I just talked about? Well, first, this has already been done when we have assumptions about the parametric case, right? And I have two references here to give to you. But the most important part here is the non-parametric case, which is the part where I'm talking about. So how can we expand this theory to take on a non-parametric hypothesis, right? And my basic solution is to forget about the parametric space and think about a space of functions, or an even more abstract space for us to dance around. Third, how do we faithfully represent the data in a non-parametric setting? And the solution I've tried so far is based on non-parametrics, right? But it shouldn't be restricted to Bayesian statistics or to Bayesian non-parametrics in particular. You can try any other strategy as long as it obeys a certain set of rules. So let's talk about pragmatic hypothesis. So we're going to do this like some sort of recipe, okay? So we have a data assumption, like X follows a specific distribution with a specific set of parameters. We have a null hypothesis about the set of parameters, which is a precise hypothesis. So we have some quite restrictive hypothesis we want to test, right? We have some sort of threshold and a predictive dissimilarity, which both will be responsible to say what is different enough from our null hypothesis and what is exactly a negligible difference, okay? Based on this four informations, we get the pragmatic hypothesis, which is basically to say all cases in my parametric space on which there's the similarity between H0 and my candidate is smaller than a given threshold which I chose. And a basic example would be to set like theta equals 0 to be my precise hypothesis, and then we're going to amplify this, right? So we have 0 here, and we're taking the absolute value and saying it is lesser than an epsilon. So we have minus epsilon here, and this would be our pragmatic hypothesis, like an extension, okay? So let's give a more practical example of the situation. And I'm going to talk to a data set which we have explored so far, which is the water droplet experiment. So it basically goes like this. We have in this upper part a cloud chamber, which is responsible to generate droplets of water, right? So once this droplet is generated, it will fall through this drift tube. And once it is in this drift tube, its position, it's registered through time, okay? So the basic objective here is to evaluate the radius of the drop, right? So as the drop falls, its radius diminishes through time. And we want to ask how this radius decreases, right? So we're going to check something called Fick's law, which states that there is a linear decrease of the radius over time. So we have a relationship between radius and time here, which is linear and decreasing, right? In order to obtain the radius, we use Stokes' law, which allows us to relate the terminal velocity of the drop and some information about temperature, humidity, and others to obtain the radius of the drop. Okay, so in a parametric hypothesis setting, we could assume, for example, that our radius is a polynomial model based on the time, and we have independent errors with a fixed variance. Okay, so our precise hypothesis, if we are going to follow Fick's law, would be that any term above beta 1, so beta 2, beta 3, up to beta p, would be equal to 0. So none of the other factors are relevant to our hypothesis, right? And then we need to derive the predictive dissimilarity function, which will say how different our object in age 0 is from age null is different from a freer version, which resides on the parametric space. Okay. In this case, we chose to use a difference in means, right? So the mean under age 0 would be this one, and the mean under no restriction would be this one. Then this will be how we would measure how dissimilar these two guys are. And based on the specifics of the experiment, we were able to recover these epsilon value for our context. So in general, our pragmatic hypothesis in this parametric case would be the set of all beta 0, beta 1s in our null hypothesis. And we're going to check these pairs here such that the maximum difference between our age 0 and candidate does not surpasses the epsilon we chose. Okay, so this would be an example for a parametric hypothesis in a parametric pragmatic hypothesis. So let's ask now about the non-parametric case. So in this case, we're going to make it a bit more abstract. So instead of saying about the parametric space, we're going to define an H here, which I'm going to just call as the hypothesis space. Okay, so we will say there is an object H star here, which resides in our hypothesis space. We have a null hypothesis right here, and then we're gonna accept H star into the pragmatic hypothesis if the dissimilarity is lesser than a given epsilon, just like we did in the parametric case. This could be translated to searching for the infimum of this dissimilarity. Okay. In most cases, we will say the hypothesis space equals to F, where F is the space of probability distributions. So we're gonna look for functions instead of looking for parameters in this case. Okay. And of course, this guy here could be quite difficult to check exactly which functions reside on it. So instead of doing that, we're going to look in a more restrictive manner to what I'm saying here to be an H line, which would be a function which is corroborated by the data, right? And we'll check if these sets of H line belong or not to our pragmatic hypothesis. Okay. Now let's talk about procedures. So first thing we have to do in order to derive these tests is to establish what kind of restriction we have on our null hypothesis, right? So we have derived so far 3 kind of restrictions, but this is by no means extensive. There may be other thought about, okay? But the first one I'm calling like hermetically restricted H0, in which the null hypothesis represents a fixed parametric family, right? So let's say I'm gonna guess that the data follows an exponential distribution with parameter lambda, and lambda can be any value that I like. So I'm not making a specific restriction on lambda here. I'm just restricting the distribution which X can assume. The second case would be what I call the functionally restricted, which in which the hypothesis represents distributions where we have a functional relationship, like the mean has to be a value, the variance has to be equal to something, the median or something like that, right? And a basic example of this would be a quantile test, We haven't yet developed this theory to its full conclusion, but we expect that throughout the rest of my studies, we may do that through functional analysis or an information theory. The last kind, which is a bit more complex to reference, is what I'm calling contextually restricted, on which I usually have more than one variable and they have some kind of relationship dependence which will be translated on my hypothesis. So maybe the most classic example would be to say, oh, I have two samples and they have the same distribution. I have the sample of X, which follows a distribution. I have the sample of Y, which follows another distribution. And in my age, know that these distributions are the same, even though I don't know which distribution is which. Then, once we establish the restriction, we need to propose a sampler for these F, right? So these F would be the way we sample from our data. We are going to do, as I mentioned, a Bayesian procedure. So we're basically updating it based on the data, get a posterior distribution, and then we build a confidence or a credibility interval for this dissimilarity here and compare it to the threshold which we choose. So this is the basic step-by-step procedure which we have developed. Right, so what we would choose as our sampler, right? In this case, we chose to go for Bayesian nonparametrics, specifically the polyetry process, which has been mentioned in the talk today by Dr. Mueller. But our basic procedure is to establish a prior, to obtain the posterior based on our data, and check if this posterior is close enough to our null hypothesis, okay? So why the polyetry though? Well, it has a series of advantages, which is conjugacy. So the posterior distribution has a known form. It is flexible since it adapts quite well to any sort of data you put to it, and it's scalable. So even if you have a large data sample, you can use parallel procedures to make the model more feasible, okay? Now, we talked about nonparametric pragmatic hypothesis, right? But we still haven't answered how we make a test answer to what the researcher wants to know. For that, we need an agnostic test. An agnostic test is basically a function of the data which result either in the acceptance, in the rejection, or in the doubt about our specific null hypothesis. So we're going to use a procedure like this. How we're going to use it? Well, we're going to associate it with the credible interval. So we calculate the credible interval between our candidate and whatever we have on the null hypothesis, right? And we have the possible results here. So let's say this is our interval. First case, if, for example, epsilon is consistently below our interval, we're going to reject the hypothesis. If epsilon is consistently above the interval, we're going to accept the hypothesis. And if it is here in the mid or somewhere in this zone, we're gonna remain agnostic about our hypothesis, right? Because in the first case here, we are consistently saying that our dissimilarity is greater than our threshold. In this case here, consistently smaller, and in this case here, it could go either way, so we rather not make an assumption. Okay. So let's make a quick review. So we have an issue that is the difficulty of interpretation. We can solve that through the use of agnostic tests. Okay. We have the issue of logical incoherence, and so far we have solved this case for at least a sub-procedure with this monotonicity, which basically states that if I have A contained in a B, right, contain or equal, if I accept B, then I should accept A. If I reject A, then I should reject B, things like that, right? So we don't, a procedure which allows us not to fall into this sort of contradiction, okay? And lastly, we have the precise hypothesis, which leads to irrelevant conclusions. We fix that through the use of pragmatic hypothesis, okay? Now, let's see how this works when we take the same example of the water droplet and bring it to this case. Now, we're going to suppose that we have the same droplet model I told you before. We use the regression procedure, a linear regression, and we obtain its residuals. And now we're going to check if the assumptions of the model are adequate, right? So we're going to test basically if the residuals of the regression model are normally distributed. This can be translated to saying that our errors here follow a normal distribution with mean 0 and a variance sigma square, where sigma is not restricted here. It could be any positively real value, okay? So in this case, we have our pragmatic hypothesis, which would be each candidate where this the similarity is smaller than epsilon. However, since we are assuming that we are on a normal distribution, this G I'm calling here will be the normal with a mean 0 and a variance sigma. And we just need to check for each sigma if there is the similarity that is smaller than epsilon, okay? So basically, for every F, for every distribution F that we sample, we are going to optimize this step. So we're going to find some sort of sigma star in which this is the least value possible and check if it is smaller than epsilon. This is the basic gist of it. So now we need to choose our dissimilarity function, right? So the objective of the dissimilarity function is to quantify what is a relevant difference. In some cases, it is more easily accessible than others, but we're going to stick here with the classification dissimilarity, which states basically that if the data was generated by my null hypothesis, we are going to do a likelihood ratio and check if it is bigger than 1, and sum it up with a probability if the data was generated by my candidate distribution, that this other likelihood ratio is greater than 1. And this dissimilarity can be interpreted as the highest achievable probability that we can correctly identify which of these distributions, G or F, my H0 or my candidate one, generated Z. So the smaller this value is, the closest to 0.5 it is, the more similar these two distributions, G and F, will be. Okay, so we have done our procedure then. We have used this function. We have found each dissimilarity for our different sample distributions, and then we have derived our credibility interval. This graph here shows an alpha here, which is related to my 1 minus alpha credibility interval, and we have here our epsilon, which says how restrictive we are going to be. Okay, and what we can conclude from this image is basically that first we have the red region, which leads to the rejectance of H0. We have the yellow region, which is the agnostic region, and we have the green region, which leads to the acceptance of our hypothesis. We can see here that we need quite a big value of epsilon. We need to be willing to be mistaken about 20% of the time, let's say, for us to be able to accept our hypothesis with a mean of alpha 0.08, which is not reasonable. And at the same time, we have quite a big restriction here for us to start rejecting our hypothesis. So in general, since for the most part, the interesting region is this one, which would be smaller values of epsilon and more restrictive values of alpha, we are mainly in doubt of our decision. This makes sense, of course, because we have just 15 observations in this data set. So remaining agnostic is more than reasonable for a non-parametric case such as this, okay? So let's wrap it up and bring some conclusions and what should be done next with the time I have left. Okay, so we can conclude that pragmatic hypothesis and agnostic texts work and are feasible. However, they are a bit more complex to implement, of course. We also remark that the predictive dissimilarity should relate somehow to the knowledge of the researcher. So it is a step that is quite delicate for us, but not impossible, okay? And as I mentioned, we haven't yet ensured logical consistency in its entirety. So the next step we're gonna do is probably to try to find intervals for F, so derive a credibility interval for the polyetry distribution instead of the dissimilarity and see what we can get from that, okay? And while there are procedures that are completely well established, like the parametric restriction I just mentioned above, the other two cases require some more analysis, okay? And of course, we would like to expand our test beyond the case where we just have a univariate variable with no covariates. The more we complicate what we are trying to analyze, the more we can say. So let's, an interesting next step would be to try to complicate our model even further, okay? So that's all I have for today. These are my main references here. But we can talk about them later. And I thank you all for the presence, the attention, and if you ever want to get in touch, these are my emails.
Thank you, Rodrigo. Any questions? Let me see. I have a question. Let me, well, I know a little bit about your work. And
yeah.
You know a little bit how I feel when you say that large samples are an issue. Just kidding. But let me ask some questions. When you say, for example, that you can be agnostic in an agnostic test, I don't know if I asked you this before, so let me ask you again. Okay, when you are in an agnostic test and you have a problem in practice and your problem falls in this region, let's say like this, in the agnostic region, what to do?
Okay, so I think these tests could be mainly thought from a scientific experiment point of view, right? So it's like, well, if you want to be a bit more greedy about your conclusions, if you wanted to try to accept a hypothesis and such, you could try this procedure. However, due to your data specification, you may not be able to do that. So I would see that as an invitation for you to rethink the experiment you did, right? And see what else you can do to make you a bit more certain about your hypothesis. So either, if this is the case, maybe I should be more restrictive about my prior, or maybe I should collect more data, or I should be more careful about the procedure which I collect the data so as to be more restrictive, right? But of course, in a day-to-day basis, I guess this graphic can be particularly illustrative in the sense that, well, you can see here what are the compromises you need to make. So you either take a bigger value for alpha, or you take a bigger value for epsilon if you want to reach a conclusion. So you can think which of these you are willing to compromise more in order to reach an assertive conclusion, of course. So if you're not willing to change the procedure or the way you collected your data, you can maybe try to think of, based on this plot, which compromise you are more willing to do and which conclusion derive from that. I guess that will be my answer.
Okay. One other thing, I'm not an expert, as you know. I don't know very well your topic of research, but by what I thought, let me state something, and you tell me if this makes sense. Okay. And in the parametric hypothesis test, you have a partition of the parameter space. And your decision basically is based on another partition of the sample space, right? You partition it, the sample space, and if your sample belongs to one part, you take a decision about the parameter space, the hypothesis parameter space. You accept H0 or you reject it, and something like that. Can you understand what I mean?
I guess I follow. Let's just see if I understood you correctly. So let's think, instead of the non-parametric case, let's think of a more simple one. Like,
yes,
we have here 2 beta 0, beta 1, and I'm trying to see if, for example, beta 0 equals 0, right? So it will lead to a region like this, which would be aligned. So my beta 1 is free, my beta 0 is not. So our pragmatic hypothesis would be basically to extend this. So we may be trying to do something like getting regions around this hypothesis and checking if, when we bring the data in, if our data comes into something that is, for example, around here for us to accept, or around here, which leads us to doubt, or around here, which leads us to rejection. I'm trying to do the parametric case because it is usually a bit more easy, a bit easier to see, but the same idea, I guess, would be valid to the non-parametric case.
Yes, my question was not specific like this, but I think your understanding is helpful to complete my question. So the threshold, the role of the threshold would be, for example, to make a thicker line to decide to take some decision about the hypothesis.
Yeah, the epsilon is basically a value which tells us how critical we're going to be about our hypothesis, how restrictive we're going to be about the things we check. Sometimes the epsilon can be fully derived, like there is this, the parametric example here, which I guess I even show you in another time, but we were able to derive that for this setting, based on the data we have and the specifics of the experiment, we could derive an exact value for epsilon, okay? So in this case, we couldn't change much of what we have. But of course, depending on the information you have, this might become even more subjective, right? Like something like, for this dissimilarity, for example, it states basically how wrong am I willing to be in the decision I'm trying to make, right? For example, if I take 0.5, which is the most restrictive case, I'm saying that I don't know where the data come from. It could have come from either of those. If it is, for example, 0.6, I'm saying that about 10% of the time, let's say, I can separate correctly which procedure generated the data. Sometimes I'm confused, sometimes I'm not. So the choice of epsilon here seems a little bit more prone to subjective judgment of the researcher in this case, okay? And there is an extra slide which I've left here if you're interested in any case, which would be how can we choose an adequate epsilon for our research? And I would say mostly it depends, right? It should reflect the knowledge and the opinions of the researcher, but if those are not so clear or so available, you could try either one of these processes here and see which is more appealing to you. These are just suggestions. You can come up with your own.
Thank you. Oops, let me see. I think we have one question here. I do not see. Let me see. It's from Mateus Piovesanotto.
All right.
I don't know if he wants to ask himself or if you want me to ask. I think I will read. Okay, Mateus. Mateus said, really nice talk, Rodrigo. Isn't it bad to introduce the freedom of choosing epsilon? Couldn't it lead to researchers perform some kind of epsilon hacking?
Yeah. I think I tend to agree with this opinion. In general, I would advise for both the alpha and the epsilon to be established before you start conducting your researcher, because of course, if you have this result in your hand, you can then think, okay, given that this is what I'm dealing with, which conclusion do I want to take, and what do I need to do to reach this conclusion, right? Which would be like this. Oh, if I choose alpha to be 0.08 and the epsilon to be 0.7, I can barely accept my hypothesis, so I'll use this. Of course, this can be worrying for a multitude of reasons. So I would say if you're doing this for research purposes, it will be better to fix it beforehand and stick to it in a way or another, right? But if you, and there are procedures, as I mentioned, to try to come up with values for epsilon beforehand. But of course, if you are in a day-to-day basis in which you need to reach a decision, you need to do something with the data you have, then I think this plot comes more in handy.
Okay. Once again, thank you, Rodrigo, for the talk.
Thank you very much for receiving me here. It was a pleasure.
Okay. Now I will pass the word to the two guys that made this workshop happen. That's Andressa and Mario de Castro. I think maybe you could say some words to finish, to close the workshop.
Pode falar, Andressa, por favor.
Okay, so I just want to say thank you for all the participants that attending all the courses and conferences in the workshop. So thank you very much for your participation. And again, thank you for all the speakers that make it the workshop happen. So thank you guys, and maybe see you all in the next event.
2 years.
2 years from now. Acho que você pode parar a gravação, Silvio.